It's a really big pleasure to be here tonight and to share, to make the big point of science, to make it wider and bigger so that everyone really understands. I'm going to talk about printable medical devices. It might look like a strange title, but it's actually very relatable to most of us. So I'm going to talk first about the problem that we are facing and then what solution we are proposing here. Okay? So when you go to a hospital, if you have a really acute problem, in most cases you need to receive some sort of medication, and in many cases you use this kind of intravenous catheter. But it might be also that you break your leg and then you have to go to the hospital because you need a screw or an implant, or it can be that you need a pacemaker. All of those are medical devices. And what happens very frequently is that those medical devices get infected. So we know roughly that 50% of hospital-acquired infections are due to medical devices. It seems that simply medical devices offer a very nice surface for microbes to attach to. We know that, for example, in the case of orthopedic implants, it's been calculated that up to 25% of cases in which you have to use revision surgery, so you have to go back to the hospital, those are due to this kind of infections. And they cost roughly 800 million euros per year in Europe. Catheters is a sort of even more serious problem. It's been estimated that there is roughly 80,000 infections due to catheters, or that happens in catheters. And the problem, it's been estimated to be around 1.8 billion euros per year. And the main issue that we have is that these infections, unlike other type of infections, are very strongly recalcitrant. That means that they are very strongly tolerant to antibiotics. Those infections then, and it's time for us to put the right name into them, they are known as microbial biofilms. How many of you have heard of microbial biofilms? Okay, that's good. It's good to be in a place where people know about microbial biofilms. So what are microbial biofilms? So bacteria, basically, let's talk about bacteria. I don't know much about other kind of microorganisms, so I'm going to talk about bacteria. Bacteria pretty much faces the same kind of decision that humans can make. You can decide to be in a Mackie in the middle of nowhere, or you can decide to live in a really highly populated city, talking to people, coming to cafes, relating to people in a very nice way. That's exactly the same thing that bacteria can do. It can live in isolation, as you see here on the top part, or then it can live in a community. And when they live in a community, in this kind of crowded lifestyle, that's what we call biofilms. If we want to be a little bit more scientific, then we can say that this first lifestyle in which they are very isolated, well, this is, we call it planktonic cells, or we talk about single cells, because they are in isolation. And what we know now is that these single cells are really the main cause of acute infections. When you have a very high fever, you have got something really kind of, for example, in your hand, something really inflamed, swollen, then you rush to the hospital. In most cases, those are acute infections that are due to single cells. Single cells are typically going around in your blood. That's how bacteria move from one place to another one. And they have been, and that's the most interesting part, they have been the focus of most of the traditional research in microbiology. All the history that we have behind in microbiology, all the many things that you probably know about microbiology, they are related to these single cells. To these single cells. Now, what happens with biofilms is that we have only talked about biofilms from the last 40 years. So we kind of only discovered that biofilms existed 40 years ago. We're going to talk a little bit about the history in a minute. They are really the multicellular state of bacteria. They get together, they want to live together, they want to be like us here. And they are really the cause of the chronic infections. Those resilient infections that don't get cured, we believe now that they are due to biofilms. And as I said, they have only been recently studied. For you to have an idea, all the antibiotics that you know, every antibiotic that you will ever get, they have been developed against single-cell bacteria. There is no antibiotic that has been developed against biofilms, at least so far. All right? So we can say that the biofilm is kind of the default mode of bacteria. We now know that really bacteria mostly live in single cells inside of the lab, but when you go out of the lab, bacteria really likes to stick to each other. We don't want to be alone. Humans don't want to be alone. Bacteria don't want to be alone either. They want to stick to each other, and we'll see why they want to be with each other. So in every surface, if they find a surface with certain conditions, they will get stuck to the surface and they will form a biofilm. So they will be in that conditions, in those conditions they will have what we call is a favorable habitat. They will enjoy the fact that they are in a community, as we'll see in a second, and that will be their default mode. They will mostly live as biofilms. So just very briefly about the history of biofilms. In the 17th century, there was a guy who actually thought that there was something funny in their dental plaque, in his teeth. So he basically scrubbed his teeth, and he sort of speculated that this dental plaque was some sort of bacteria. He thought. He was actually quite right. Then Pasteur, who you probably know very well, he speculated that aggregated bacteria was actually the reason as to why the wine will go bad, will kind of it will taste bad. Then Henry Shih actually was the first guy who realized that there was some sort of structure, aggregated structure, when you would put glass slides in water. So he saw that there is something floating around in water that might be bacteria. But it was not really until the 1970s, as I said, that the term even was coined for the first time. And it was due to the work of William Costerton. He's considered the father of biofilm research, of modern biofilm research. And in Europe, this was an American scientist. And then in Europe, it's very widely recognized the contribution of Niels Hövi. He's still alive. He's doing a lot of research. He was the one who basically established the association between biofilms and cystic fibrosis. So you probably know cystic fibrosis. One of the big problems in cystic fibrosis patients is the fact that they have very recalcitrant infections in their lungs, which are due to biofilm bacteria. So how are biofilms structured? What are they? When you have a biofilms, these are pictures that we've taken in our lab. You have cells, these are the cells here, and those cells are really interacting with each other, and they are embedded in something that we call extracellular polymeric substances. You know, scientists, we like to make fancy names for things. You can also call it matrix. They are really embedded in something that we call matrix or EPS. And mostly occur attached to surfaces, not in all cases, but biofilms are mostly related to surfaces. And a very important thing here is that the biofilm, the main feature of the biofilm is the presence of the matrix. If you were to destroy the matrix, those cells will get loose and they will immediately switch into the single cell lifestyle. So the most remarkable feature we can say of biofilms is the presence of the matrix. So why, we've been talking about it, but I wanted to come back to the analogy of the humans and the bacteria. So in a community, in a biofilm community, basically you have biofilms living in an encased structure in this matrix. It's pretty much the same thing that humans live. We live in buildings, we don't live out there, so we want to be protected. That's exactly what bacteria do, they want to be protected. We humans communicate, in most cases we talk to each other. That's exactly what bacteria does. They produce signaling molecules that sense different things to the neighboring cells. That's known as quorum sensing. If you ever hear this, it's the way that bacteria talks to each other. Then we also know that if this will come back, that will be nice. Yeah, then another important thing is that in a city like Helsinki, we share cafes, we share streets. That's pretty much the same thing that biofilms do. They share nutrients, they share their structures, they share signaling molecules as well. So this cooperative community really makes bacterial life easier, and that's why they want to live as biofilms. So this is how biofilms look like. As a scientist, I think you always want to look at the things, even if you have indirect evidences. So this is Staphylococcus aureus biofilms here. This is Pseudomonas aeruginosa biofilms. This is the biofilm that forms our dental plaque. It has a very long name. It's no point to pronounce it. But those are like pretty much how biofilms look like. Here you see in green cells that are alive, and in red you see cells that are dead. So they get killed, they get flushed away, and then the living cells continue to produce or to multiply in the biofilm. So And I apologize for these disturbing images. So there are two main problems associated with biofilms, the challenges of so-called bacterial biofilms. The main problem is that, as I already told you, they are very recalcitrant to therapy. It has been estimated that to kill a biofilm, you need roughly 1,000 times more concentration of an antibiotic than if you compare to a single cell. So this is an example. This is a lung of a patient that was treated with antibiotics for 20 years. He had cystic fibrosis, and when he died, biofilms were still alive there. So it didn't matter that you treated the patient for so many years with antibiotics, biofilms were still there. The second big problem that we have is that typically in a healthy human, if you are infected with a single cell infection, kind of infection, the immune system is really good at clearing out infections. It recognizes the pathogen, it clears out the pathogen. But that's not the case with biofilms. So here you see a single cell. The blue part, it's an immune cell. It recognizes the pathogen and it just clears it out. It removes it from the circulation. It eats it, and then it removes it from the circulation. But that's unfortunately not the case for biofilms. For biofilms, we don't really understand, or we don't understand all the molecular details, but we know that immune cells come to the biofilm is really here in red, they come to the biofilm and they get really paralyzed. They can't do anything. They are just somehow affected by the presence of the biofilm, and they can't go any further. So we say that the biofilms create an immunosuppressive environment that then really makes it impossible for the immune system to recognize that they are bacterial cells and to kill this structure. So we know nowadays that biofilms cause a lot of problems in device-related infections, as I just said, all kind of medical devices associated infections, but they are also responsible for other kinds of infections that are not associated to medical devices. For example, otitis media, it's one of the most common diseases in kids. It's due to biofilms. Well, I told you already about I told you about cystic fibrosis, but also there is urinary tract infections. There is a number of infections that are due to biofilms. So biofilm research has only happened, as I told you, it was discovered 40 years ago. There is a really huge need for more research on biofilms, but it's really nice to say, and I'm really proud to say, that the community in Finland is growing. We have, last January, we had the first meeting for all the biofilm researchers in Finland. There is research in the area of environmental biofilms, in the area of medical biofilms. So we hope that more and more we will be contributing in the area of biofilms. What brought me here, though, is that we seem to have a solution for this problem, and it was our idea is that for building better medical devices that will be able to resist infections, we can use 3D printing technologies. A couple of years ago, we had actually shown that if you print a disc, small disc, and you incorporate an antibiotic into this disc. This is the disc that I was telling you about. This disc with this polymer that was printed with the antibiotic inside was able to protect better, to resist better an infection than a simple disc that would have the antibiotic just added on top. So we basically realized that by using 3D printing as a delivery method, we could actually have a medical device that would be better protected against infection. There is also a very important thing about 3D printing. 3D printing is revolutionizing our life, among other things, because you can customize things. You don't need to do things differently. You don't need to do things in the same way. So we are all different. So why can we assume that all the medical devices would be the same way? We are different. We don't have to use the same catheter for a small person that we would use for a big person. So we can customize the medical device according to the needs of the patient, and in that manner we can prevent all the problems that you can have with medical devices, such as inflammation and remodeling, tissue remodeling issues, and so forth. So then we had this idea that if we use 3D printing technologies with good antibiotics, then we can have better medical devices. Then we applied from the European Union to get to obtain funds to develop this project, and we ended up having last May we were granted 2.3 million euros, and this is a European training network that involves 9 PhD students. So 9 PhD students will be trained in this network from next year on, and they will be exchanging, or they will be moving around 6 European countries and also the US. And there are 2 companies companies that are involved in the project. So the students will be receiving both academic and industrial training, which is a big a big objective of this kind of European training network. And well, it says there that I was granted this project last May, as I said, but at the same time, or it was very shortly after, I moved to a different position. So I'm currently not working directly for the University of Helsinki anymore. So Professor Pia Vuorela is now on the coordinator of this project. So if you have any doubts in the future, you can contact her. There is a picture of her here. And our project manager is Dr. Karita Peltonen. So the University of Helsinki will continue to coordinate this project, and I will be involved in the supervision of students and also in the advisory board. So what we want to do with this project is that we want to identify and characterize really good compounds that can kill biofilms. We already have some of them, so it will be just building on this knowledge that we have already obtained. With those ones, we are going to develop 3D printed devices and formulations, and because we cannot do everything in 4 years, we are going to focus on 2 specific devices. It will be an endotracheal catheter and a femur implant. So we hope that at the end of the project, we will have prototypes, really ready-made prototypes of both implants, and that we will be able also to test them in vivo models. And we want to do this in such a way that we show that it's possible to integrate in vivo with in vitro data. We are going to share this information that we have obtained when the time comes, when the time is right. But we are going to do this in such a manner, that students get trained in all the different stages of the drug development process. So our goal really is that we teach them not only how to develop new compounds, how to make new biofilm devices, but medical devices, but we also train them in how to exchange from academia to industry, how to get a set of tools that will allow them to transfer skills, not only scientific skills, but also skills that are needed in nowadays world. So we really hope that this will be very beneficial for the students. And with that, then I end up this talk, and if you have any questions, I'll be happy to answer. Thank you.